I came into this with basically no lab experience, which is kind of the whole purpose of why I wanted to do iGem. Just thinking about grad school, I get to see, new people that have the same interests as I do and are so brilliant and hardworking and more knowledgeable than I am. So I've learned so much from them.
Working with the iGem team has really been a great experience for me, and it really just encourages me to work out with others and to just make everything more of a team effort. There's many different benefits of working as a team. You get more ideas and generally just quicker protocol, just finding information, everything just happens to come together better when you're working with other people.
I can learn from how other people do things and kind of pick up on little tricks and stuff that they do that makes them successful in terms of carrying out protocols for one thing, and also just how they think and how they approach problems. So you can pick up stuff from other people and you can use that, use the stuff that you've learned when you want to do your individual research.
We had some very good initial brainstorming exercises to come up with a project, and that was very interesting. And it's exactly the kind of thing that I thought iGem was going to be all about.
And it was really a consensus sort of way of figuring out what we wanted to do. And then when we started the summer, we had our broad topic of bacterial targeting, but the subtopics, the sub-projects were still evolving. And really it came down to what students wanted and what students found interesting and said, okay, here's what I think, and here's what I think would be really interesting to sort of test and see if we can make this work.
Our project had 3 sub-projects: quorum sensing, cell targeting, and effect signaling system. And it's really interesting how, there were a couple people in each and everyone had a little bit of a different interest, but through that we were able to kind of thread together a lot of different ideas to create this big project that I think really works well together, and I think we're really all excited about for the Jamboree.
And for example, our project, the Effect A project, just happened because we were saying, okay, are there any signaling pathways on the outer membrane that we can sort of hijack? And we started looking, and we did literature searches upon literature searches, and we finally found this one. We said, well, hey, maybe this might be worth a shot. And the faculty were completely supportive, and the postdocs and the RTFs were completely supportive, and they said, all right, if this is what you want to do, go for it.
And so far it's been a great experience. I've learned a lot during this past summer. During the semester I was working in a microfluidics lab for this postdoc, and most of the time it'd be just me going in some afternoon, and I might get some instruction from the person I was working for, but most of the time I'd be working by myself, and it'd be kind of lonely, and also there wasn't much interaction with other people that were working on the same thing. But for iGem, every day basically, I come in the morning and there's 6, 7, 8 people there working on pretty much the same thing, or at least related topics.
And that's another part, it was really presented, and it has been something that is an undergraduate project, not a professor's project or the postdocs who are just basically using us for mini prep after mini prep. We're able to sit down and say, well, what direction do we want to take and what do we want to explore? And I think that's been really one of the best things about iGem.
It was a much more friendly environment. I could approach people, I could ask them about things, and a lot of them were really knowledgeable, and also we had TFs there that were there all the time, so I could always find people to answer my questions.
I've done work before, and all this experience that I've had, I've been able to carry on and pass it on to new students, and I've been able to teach them everything I've learned, and hopefully they'll continue working, learn even more, and teach them and pass that on to iGem 2008 and years later.
This has certainly been a good experience and a useful one. I'd recommend it in particular to people who are interested in biology and have some research background and who are self-motivated. Those are really the people for whom iGem's a perfect opportunity for. Even then, if you're interested in biology or synthetic biology, this is a really great position.
Everything starts with the students, and that's what I really enjoyed about that. Everything that we thought of, the concepts and the actual experiments and the meat of what we were doing was created by the students, thought of by the students, executed by the students, and finally presented by the students at the end. And it's really becomes our own thing, and being able to have an ownership over our work and our project is wonderful, and being able to design everything ourselves is fantastic. But we're also not left alone.
We have plenty of faculty advisors, which is so attractive. Like, a little intimidating at first when we walked in the interview, and there's Professor George Church, and there was Professor Deborah Auguste and Pamela Silver. So some really great names at the forefront of synthetic biology who were willing to work intimately with 8 undergraduates.
iGem has given us the chance to kind of figure out what we want to do by ourselves, which is really good and kind of misleading in terms of what lab work I guess seems to be, but it's good because that's what my experience of lab has been, and hopefully what my grad stuff will be. I've
come out of this with the knowledge that yes, I can do this.
I think one of the greatest things about iGem is it's really student controlled to an extent I would have really never thought possible. So it's great that we have the TFs there to help us along, but we still get to make our own mistakes, and I think in a lot of ways just the whole freedom of it allows for it to be a richer experience.
If I had an idea that I, as an engineer, I thought could, was workable, but then I didn't have the exact details, and, talking to someone else who had done MCB, for instance, molecular and cellular biology, was very helpful in that I was able to actually whittle down to the most essential parts of the project. The interdisciplinary setting also helps in that you get to see how somebody who studies something else totally different from yours has a certain way of thinking.
It's very interesting, especially since I'm kind of a math nerd. I do the engineering thing, so it's kind of cool to have that whole computational side and be able to use that for a biological purpose. We've talked to a whole bunch of other people who've worked with the system that we're using, gave us all kinds of tips on what kind of media to grow it in, what conditions, what cell dilution. So it's just been really like a huge collaboration of different groups, and it's definitely enriched the experience to have all those different people give their input. Sometimes, different groups do different things. I mean, we'll all be working with the same system, and one group will say that this media works, and for some reason that media doesn't work for us, so we try other things. So, I mean, it also gives you the perspective that there's so many different ways to solve the same problem.
Science takes time. It really does. You think that, okay, we're going to test our hypothesis, and when you get into the lab, you figure out you have this great scaffold, this will work perfectly, and you think, okay, next week I'll be ready to start testing out my hypotheses, seeing what's going wrong, and start troubleshooting. And that's not the way it works at all.
Then the whole experience has been very enlightening for me. I've actually seen how very powerful a tool it is, the ability to create genes.
The key point of synthetic biology is that it allows us to use cells to manipulate small molecules, and that's something that we have a rather terrible time doing with on a macro scale, but we can use cells as machines to do it for us.
Everything is novel and everything is new, and so everything that you come up with is your own thing and a new thing. And usually it builds on something that's been done before, but the ability to use synthetic biology to put together parts, it's a really cool idea.
I would love to continue research, and iGem has made me really excited about having my own project. So even though I'm only a rising sophomore, I'd really like to continue to do my own research, and in a way, continuing the iGem research is almost a means to do that.
I will definitely, no matter what I do, I'll take away from this experience the independence and sort of the realization on how progress actually happens, and that is really through the will of people willing to stay there in the lab and willing to stay there and say, okay, I believe in this and I'm going to do what it takes to see it through.
Trying to take biology and turn it into technology, that's I think my biggest interest, to take biological knowledge and be able to churn out tools that can be used in research or therapies or some similar endeavors. And I hope to be able to use some of the techniques that I've learned in the lab this summer to answer some of the questions that have had very elusive solutions to them.